joined now for this little bit. So I'll share my screen and I'll pull up the big idea. There we go. Whoops. I don't know why I just closed it. Oy. All right, there we go. Okay, so is there anything on here that you want me to do first? What do you want to go over? What in this unit do you think is hard?
Can you do the operons and stuff?
Actually, it's a little bit easier for me if I share a whiteboard. It's just way easier to write on a whiteboard. I know it's sort of small, but it's hard to write on a Word document. Okay, so basically remember this. Circular DNA is found in prokaryotic cells. So if you have a bacteria, here's the whole bacteria. This is its one circular chromosome. So it still looks like this. It's still DNA, but it goes around in a circle. It's a circular piece of DNA. And that for things like bacteria, they have one main chromosome, that's their circular chromosome, and then they have little extra circular chromosomes, and those are the plasmids. And the ones that they tend to exchange are these ones. They will let them out and they will pick up others. And we know for an absolute fact that that's how resistance gets passed from one bacteria to another. So when we start worrying about like, oh, all of these bacteria are now ampicillin resistant, we got to come up with a new medication. That's why they did what's called transformation, and they shared their plasmid DNA with the environment. Okay, so now I'm going to go to this main chromosome. This is my main circular chromosome. There are literally over 1,000 genes in most bacteria. So I'd have to divide this into 1,000 pieces. So now I'm just going to show you, all right, I have one of those pieces, one gene. So the gene is down here. We call it the structural gene. That's like, all that means is like, what thing am I really coding for? So let's see the structural gene. Everything codes for mRNA because that's what DNA codes into. Transcription gives you mRNA. But the mRNA specific to this one, let's say at the ribosome, it builds lactase. Lactase is the enzyme to build, to break lactose. Now, you do not need this all the time. So this is a gene that's normally off. And now I'm going to explain how an operon works because I'm going to explain how it's off. So the lac gene is actually made up of multiple parts. It's got this part that we just talked about. Ahead of it is the operator, which is like just an on-off switch. Ahead of that is the promoter. The promoter, you always remember because we did it in class with our piece that we built, you had to find the Tata box. That's where transcription begins. So the Tata box is where my RNA polymerase will bind, and it's going to start doing transcription in this direction, as long as it's not blocked. So that's what I'm going to come to next. Ahead of that, so when something is ahead of a gene, they call it upstream. So upstream of the promoter is a regulatory gene. And if you did the Unit 6 FRQs, I'm sorry, I have been really, really busy, so I didn't get to grade any of the FRQs that you posted, but I promise I will go in and grade those. And now that we're on Unit 6, I'm really focused on everything FRQs from here on out, making sure that you're used to everything. So I apologize that this was a bad grading week for me, but I will go back and grade things that haven't been graded. But anyways, regulatory gene upstream. The regulatory gene makes mRNA that goes to a ribosome and makes a repressor protein. Repressor proteins bind to operators. So I'm going to draw a little arrow. It goes up here. I'm going to make it just be a different color so it shows up. There's my repressor protein. Now it looks different from my RNA polymerase. If this is in the way, my RNA polymerase can only get to here and then it's stopped, which means I'm not making mRNA, which means I'm not making lactase. So that is why this gene is off. Does that make sense? Okay, now because it's super messy, I'm gonna go like this. Here's my gene, promoter, operator, structural gene, regulatory gene, repressor protein, and RNA polymerase. Oop, RNA polymerase. Okay, so right now, this repressor protein is blocking the way. So transcription's not happening. The thing that will make the RNA polymerase get out of the way and go down here and get out of the way is the presence of lactose. So lactose, I'm drawing it as 2 circles because I know it's a disaccharide. When lactose is present, when that substrate is available, the repressor protein likes the lactose more than the DNA. So it moves out of the way. If it moves out of the way, this guy's sliding along. If he's sliding along, he's going to make the mRNA to go to a ribosome and make lactase. Lactase is the enzyme that fits this and breaks it apart. That way, this operon is what we call inducible, meaning you can induce it. You can turn it on. Normally off. Hang on. More delivery. Sign. Okay.
And I only have a 20. I'm sorry. I go to the bank because I didn't have a 20.
I will figure it out. Hang on one second.
We haven't reached
I
out
will
to buy
do
one.
it
If you
Zoom
don't-
with
if you
my
reach
class.
out- Oh, I'm sorry. No, it's my class.
Hi, class. Bye over. Okay. Is that
If Tracy reaches out, I'll let my
okay. So 10 and that's 3. Okay. That will $1 goes into the perfect. Yep. Sorry. Oh my God. There's so much going on. Okay. This is inducible because you could turn it on, but there's no sense for bacteria to make lactase unless it actually needs it. So that's why it's inducible. Here is a human cell, nucleus, DNA, tons of DNA in here. This is not controlled by operons. Instead, this is controlled by transcription factors. Transcription factors are specific proteins that are made due usually to a ligand, and so that transcription factor will go and bind to a very specific gene and say, okay, RNA polymerase right here, this is where I need you to work. And RNA polymerase goes, okay, I need an A and a U and a C and a C and a G. Okay. There's 2 kinds of operons. The one that I showed you was inducible, which means it's normally off, but you can turn it on. The other type is repressible. Luckily, these words mean exactly what they stand for. So repressible is normally on, but you could turn it off. So it makes this product, and the product activates the repressor protein and turns the whole thing off. So if you make too much, you can turn it off. So both of these operon controls are what we call feedback. Feedback means that if you get not enough of something, you can now turn it on and make it. If you have too much, you can turn it off. So both of them are feedback mechanisms. What else? So both of those control transcription, or I should say all 3. Operons and transcription factors, and both operons, repressible or inducible, they both control transcription.
Can you do like the conjugation and transposon?
Yes. So those are words that I really didn't expect to see, and they somehow just slid them on there. So conjugation is if two cells actually direct contact exchange DNA. So they call it bacterial sex. Bacteria do not normally do what's considered sex, like they do not do sexual reproduction, they don't make sperm, they don't make eggs, they don't have fertilization, but they can direct touch and give each other DNA. So that direct touch and give each other DNA is called conjugation. If instead, here's one DNA, or here's one bacteria, here's another bacteria, and I let out my plasmids and you take it up, that was called transformation. That's what we did in our lab. That's how we got E. coli to take in the glowing gene and take in the ampicillin resistance gene, transformation. And that word originally came from Griffith, the guy who noticed that the R bacteria were letting something out, or I'm sorry, the S bacteria were letting something out and the R were taking it in. And that when they took it in, they got a capsule that looked just like the S. So he called it the transforming factor, and now we call it transformation. Was there another word you said? Oh,
It was the transposon.
Transduction. Oh, transposons.
Yeah.
Yep, two more. Transduction is when a virus gives something to, so in a virus, you can have RNA or DNA, like COVID is an RNA virus. It's a retrovirus, like the HIV viruses. So when you get it from a virus to a bacteria, that's called transduction. So whoop, it gets inserted. We definitely did that this year. We talked about how a virus will insert DNA into a bacteria. So that's called transduction. And the last one is called jumping genes. And they're not necessarily jumping to a different bacteria. If this is my bacteria, it can actually take a gene that's here in the main circle and here in a plasmid and move it. So jumping genes, transposons. It just means they'll move. It doesn't happen very much in humans. I know it has been recorded to happen in corn. A great example is it will move from, like, in corn, it'll move from chromosome 5 to chromosome 8. That's a jumping gene. It used to be in chromosome 5 and it actually broke off and moved. So all of these things, transposons, transduction, conjugation, and transformation, they all increase the genome. They all increase the variety of genes that are found in any organism. So if you compare, like, bacteria over time and you're like, wait, why is their DNA changing? I wouldn't expect bacteria's DNA to change because they don't even do sexual reproduction. Well, it's changing because they'll take things from their environment, they'll get stuff from a virus, some of their own genes might move. That's how variety happens in something that reproduces asexually. Because I feel like in baby bio, you probably only learned that they're, like, really not variety. But there are some ways that there can be variety in prokaryotic cells. Next, unless you want more on those. I do apologize. That was by far the longest video, but they are not evenly divided. There's just way more content in Unit 6 than Unit 5. What else? So I'm going to go right to AP Classroom. I can go there. Okay. You guys still see my board when I change it? Like, does it show me signing into AP Classroom? No. All right, I'm gonna have to get to it and then share it again. If you haven't tried submitting the FRQs for the unit, I think I left them all open. I would definitely do them even if I've gone over the answers, because it just gets you to practice like how this works. I mean, it's just a little bit of a slow process to get there. Okay, can everyone see this? This is the actual, they call it the scoring guide. So like, that should look familiar to you. That was the first question. So let's just back up a little bit. And it says, CD3 is a signaling protein that's typically found in the plasma membrane of immune system T lymphocytes. CD3 is composed of several different polypeptides, including a gamma chain, CD3 gamma. Scientists analyzed the promoter of the CD3 gamma chain gene for regulatory sequences that might have positive or negative effects on the expression of the gene. Okay, so we stop right there and we digest it. Okay.
We can't see the screen, Mom.
Oh, you can't?
No.
Now can you?
Yeah.
Yeah.
So I'm not on AP Classroom. I actually downloaded something from AP Classroom because I can move it easier than when I'm in AP Classroom. Of course, it took me 6 units to figure this out, but I figured out how to download it. So here was the question. So the first part has just told me that a signaling protein that has multiple parts to it could have regulatory sequences that are positive or negative, which means they're going to actually turn on or turn off the gene. And if CD3 is a signaling protein, it makes me think like of a transcription factor or turning on a transcription factor. So that's what I'm thinking about so far. The scientists cloned fragments of the CD3 gamma gene that included the first transcribed nucleotides, plus up to 789 nucleotides of upstream regulatory sequences. Okay, so here is my gene, and they're saying, okay, we found all of these things ahead of it, upstream of it, and regulatory means that they can make like repressor proteins to turn something off, or even something to maybe turn it on. All right. It says they cloned them and they put them into plasmids. Yay, I know what that is. They're telling me they took all these sequences and they put them in little circular pieces of DNA that could be put into the environment, that we could heat shock our organism and have it taken. All right. The plasmids were then introduced into a line of T lymphocytes, and the cells were allowed to grow for a short while. Because the regulatory sequences immediately precede the luciferase gene, the activity, either positive or negative, of the regulatory sequences affected the amount of luciferase gene expression by the T lymphocytes. Luciferase catalyzes a reaction that results in the release of light and is responsible for bioluminescence of fruit flies. By quantifying the bioluminescence in the cells, the scientists were able to determine the effects that they cloned. Okay, so I want to make sure when you read this that makes sense. So upstream, so this first little picture here is showing transcribe region is what has to be transcribed. Upstream regulatory is telling me sequences that could turn on the gene or turn off the gene, because maybe they make repressor proteins or maybe they make transcription factors that are activators. Either way, this is a eukaryotic cell because it tells me it's a lymphocyte. So it's not like an operon, but a regulatory sequence is transcription factor that's on or off. It's a repressor or it's activator. Then it shows this plasmid. So it shows this small piece of DNA that they're getting the T lymphocytes to take in. And so you might be thinking in your head, wait a minute, plasmids are for bacteria. Plasmids were first found in bacteria. But if we could get human cells to take in plasmids, we can alter human DNA as well. So that's the research that they're doing. They're trying to get your T cells. So like you've heard of killer T cells maybe, or helper T cells, like those are immune cells. A lymphocyte is an immune cell. So they're trying to get you to take them in. So if they put the glowing gene right after the sequence, they'll know, because if you read it all and then you eventually got to the luciferase gene and glow, you read it all. So if you glow, you did it. It works. It's sort of like how we had the ampicillin resistance gene in there. Like we knew that if we put it on ampicillin and it survived, it got the gene. It worked. For them, glowing is letting them know it works. Okay, so then the questions. Oh no, not the questions yet. The CD3 gamma upstream regulatory sequences cloned immediately before the luciferase gene were constructed. Each type of plasmid was introduced into T lymphocytes. So that's also telling us that they might have actually put it into the cell. They don't say if like the cell took it in or they injected it into it. But the amount of luciferase produced was dependent on the regulatory sequences present. So if I look at this quick, I look down here and go, all right, the parent plasmid made the least, and this one down here, the parent plasmid with no promoter on it, made the most. So I have this different range of how much luciferase, how much it worked or didn't work. All right, analysis of the luciferase activity containing plasmids with different amounts. Names of the plasmids are shown on the left, included regulatory sequences are shown on the right. Error bars are plus or minus 2 standard errors, and the transcription start site is indicated by +1. All right, identify both the cellular component and the location of the component that is responsible for producing the luciferase protein. Okay, so what makes the protein? What cellular component? What were they looking for an answer? Where are proteins made in a cell?
The ribosome.
Yes, that's what they were looking for. They were looking for ribosome. So when it says identify both the cellular component is the ribosome. And then the location of the component. Where is a ribosome?
Cytoplasm.
Cytoplasm. Yep. Because if it's going to do something right in that cell, it's going to be right in the cytoplasm. So that's what they were looking for. And then the next thing says, explain what dictates to the lymphocytes the correct order in which amino acids should be linked. What gives it the order?
The mRNA?
Mm-hmm. The bases in the mRNA, like the order of the A's, T's, C's, and G's will determine, oh, sorry, no T's. This is mRNA. A's, U's, C's, and G's. The order of the A's, U's, C's, and G's make up codons, and those codons call for tRNA anticodons to come in. They carry that specific amino acid, and that is what determines the primary structure, which is the correct order of the amino acids in the luciferase protein. Yes? Okay, next one. Identify the independent variable in the experiment described. What is the one thing that they change Independent variable means one thing that they change.
The amounts of CD3 in the regulatory sequence.
Yeah, the amount of regulatory sequences in the plasmid. The amount of, so over here, sorry, I touch on it, but you can't see what I'm touching. Oh, you can if I use this. This is what they're changing. They are changing the amount of regulatory sequences. That's the independent variable. All right, let me go back to the question. Oh. Identify the plasmid that was used as a negative control for luciferase activity. Justify including the plasmid with the not justify, oh wait, sorry, one at a time. So independent variable was the amount of regulatory sequences included. Identify the plasmid that was used as a negative control. I feel like they are 100% going to ask you for a negative control in your question. In your, like, you only get 2 questions on your exam. The first question is going to be a lot like this question, where tons of, not tons, but like at least 2 pieces of data. And then I would say at least 5 parts to it, A, B, C, D, E, or multiple parts to the, like right now we're on B or C and there's 3 parts to B or C. I feel like it's going to be like that, so they can maximize the amount of things you could potentially write about. So what's a negative control? Negative. Showing that it's no. Where do you see no on here? Which one doesn't work?
Parent plasmid?
You got it. Parent plasmid. That's the negative control, the parent plasmid. It doesn't have any regulatory sequences added to it. It doesn't have any extras. And then the next part of it says, justify including the plasmid with the non-active promoter in the experiments. Justify including the plasmid with the non-active promoter in the experiments. Why would you include this one, the bottom one? Why do you include this bottom one? It shows how much luciferase activity could happen if you don't have any regulatory sequences. So now I'm just going to scroll down here because they show you, this is how they actually grade it. So part A, the response indicates that luciferase is produced by ribosomes in the cytoplasm or the cytosol. Yep. The next part, the sequences of nucleotides in the mRNA, so the order of the A's, U's, C's, and G's, dictates the amino acid sequence of luciferase. The triplets called codons specify each amino acid to be added in order to the growing polypeptide chain. Yes. The response indicates that the independent variable is the particular plasmid or that it is the CD3 regulatory sequences. Right, so we were saying that. And then the negative control was the parent plasmid because it didn't have any regulatory sequences inserted. And then the plasmid with the non is a positive control for the effective introduction of the plasmids and for the successful use because it had the greatest amount that it made. All right, and then the next one, oh, I don't think we read the next one. Okay, this is the next question. Identify the plasmid that must contain the CD3 gamma core promoter sequence, but the fewest or no regulatory sequences. So that's the first part. Which one is going to have the least regulatory? So I'm hoping that in your head, you're just muted, but in your head, you're like, okay, if it has the least regulations on it, it's going to make the most, but I can't pick the bottom one because that has no sequences in it. I have to pick one of these plasmid ones. So I'm picking this guy. Oh, I got to use my cursor so you see what I'm putting at. I'm picking this guy. This one made the most. So this one is called PCD3 gamma-15. That one. That must have the least regulations on it because it still made the most glowing protein. So it wasn't stopped. All right. Then the next part of the question says, based on the data in figure 2, describe the most likely cause of variation in luciferase activity amongst the cells in 419, 309, 239, 199. Okay. 419, 309, 239, 199. What is different about them that makes their results different? They each have different what? So whenever you're not sure what it has, look underneath the figure because chances are the answer is going to be right here. So the difference between all of these plasmids is that they have different amounts of regulatory sequence. So that's why they're different. That's what you have to put. They have different amounts of regulatory sequences. Then the next part of this question, actually I'm really, really liking that there's 3 questions inside a question because it makes me not so fearful of the whole, there's only 2 questions on the exam kind of feeling. They're at least asking you a lot of questions within questions. More chances to get things right. All right, calculate the approximate % increase in luciferase activity between the cells containing 59 and 149. Okay, let's go see what the difference is. 59 is right here, so I would say that is around 2. And then 149 is right here. I would say that is 4. So if this one makes 2 and this one makes 4, you have now doubled the amount. So that's 100% of an increase. It has increased the amount by 100%. If you go from 2 to 4, that's a 100% increase. That's like something costs $20 and all of a sudden now it costs $40. If something doubles in price, it has gone up by 100%. All of its value increased by that amount. Yes, that makes sense? Okay, so let me just go down to their little scanning things like this and see how they wrote these answers. Okay, this is the first one. It says the response indicates, and by this they mean your answer, indicates that the plasmid must contain the core promoter sequence, but with the fewest or no regulatory sequences must be the 15 because it gives the greatest activity. Okay, we said that. And then the next one, the response indicates the different sequences cloned into the plasmids contain different combinations of negative and positives. Yep, so it's the differences in the regulatory sequences given. And the next one, the response indicates that the percent increase is 100%. Yep, because it goes from a 2 to a 4. Okay, now part D. Predict the most likely observed level of luciferase activity if 789 is introduced into non-lymphoid cells, such as a line of kidney tissue cells. Provide reasoning. All right, any ideas? I know my guess. If you put it into a different cell than a lymphocyte cell.
Wouldn't it just not be able to like carry out its reaction or like the glow?
Yep. Yeah. If you use a different cell, it's probably not going to work because it doesn't have the same transcription factors. It might not have the same signal transduction pathway with the right receptors to give you the transcription factor. The transcription factors are what's going to control your regulatory molecules and if they work or don't work. So I would say you go trying to put it in another cell, it's not going to work. Let's see how they wrote it. I just pulled up these documents, so I'm not sure how they write these. Oops, I've gone too far. Oh, all they put was the response indicates that non-lymphoid cells will not contain the transcription factors that bind to the regulatory sequences. That's it. That's all they were looking for. They're not, but knowing that that's all they took, I can be pretty confident that if you write that it, because they don't have the same receptors, they wouldn't lead to the same result and wouldn't produce the same transcription factors. If you go about it that way, that also I would say would be correct. All right, any questions from this first one? I like that they showed you it circular like this, this plasmid that they were putting in there, because I know those are, they look like what we did with our gene. I think that any of these diagrams that look overwhelming when you first look at it get easier as you read about it. Take the time to make sure you read what's under it and you understand what this piece of information is actually telling you. All right, I'll move on to the next one. Okay, question 2. So question 1 is always going to be based on some experiment that they did, asking you about the negative control and asking you about why it worked the way it did. What would happen if it didn't work? Or what would happen if you disrupted it? What specific data points are telling you? I feel like those are exactly the choices that are going to be your A, B, C, D. All right. The second question is going to be more content related, not experiment related. So this one says DNA replication in eukaryotes and prokaryotes starts when a complex of proteins binds to an origin of replication and separates the two strands to form a replication bubble with replication forks at each end. In order for replication to proceed, the enzyme topoisomerase binds to the DNA immediately in front of the advancing fork. I'd be psyched like crazy if this came up because you literally all built your own replication forks. Like, you know this stuff. So there's our origin of replication, meaning that's where it's gonna open up. That's where you're gonna start your little bubble. You're gonna have a fork going in each direction. A, describe how the structure of a prokaryotic chromosome differs from the structure of a eukaryotic chromosome. Give it to me.
Prokaryotic is just one in a circle.
Yes. Till they get all the questions right. Yeah. All right. Yes, you're going to put prokaryotic is circular chromosomes, eukaryotic is linear, just like straight chromosomes, lines. Explain why DNA replication cannot proceed to the regions of the chromosome labeled as I in figure 1b unless topoisomerase binds ahead of it. Why can't it get to here without, what does topoisomerase do, is basically what it's asking you.
Doesn't it like hold it open?
Single-stranded binding proteins hold it open.
Oh, that it unravels it.
Yes, perfect. It unravels. DNA is super coiled, like it's all around each other. It's got all those nucleosomes and it's trying to fit in a really small space. So if you don't unravel it first, it's very difficult for a helicase to get working. So good, unraveling, perfect answer. All right, the next one says, Use the template of a replication fork to draw arrows that represent both continuous and discontinuous DNA synthesis. Draw one long arrow to show continuous DNA synthesis and 3 arrows to show discontinuous. The arrows should point in the direction of the nucleotide addition, and the 3 arrows should be numbered 1, 2, 3 in the order of fragment synthesis. Okay. I gotta go like this. You see my whiteboard now? Oh, you don't? All right, do you just see the whiteboard?
Yeah.
So if this is what they gave me, and they put, sorry, I gotta go back and look. 5 and 3. Where'd my whiteboard go? Oh my gosh. Okay. They add a 5 here and a 3 here. And so they want you to draw a continuous, which is your leading strand, and they want you to draw 3 Okazaki fragments to make up your lagging strand. So first thing you have to tell me is, what is the direction in which all fragments are built? All DNA is built in this direction.
5 to 3 prime.
5 to 3. So that means 5 to 3, this one is going to be my leading strand, my continuous segment towards the fork. Then I'm going to make 3 pieces like this. 1, 2, 3. They're all going 5 to 3. They're all going 5 to 3 because they're all being made away from the fork in discontinuous fragments. Which one? I'm going to hold my little cursor. Is this number 1 or is this number 3? Do you see my cursor or no? Is this built first or last?
Last.
Got a zipper. Well, maybe it's easier to answer this. Where's the next one going to be built? Over here or down here? I have to unzip my DNA, right? So my next piece would go right here. So this was built first, this was built second, this was built third. My next one will be here. This will keep unraveling and I'll make another one. And then I'll unravel more and I'll make another one. And it'll keep going that way. So they're made in discontinuous pieces away from the fork. You got to think about this whole fork at the very beginning looked like this. It was hardly unraveled. So there was only piece number 1 that came first. And the more that it unraveled and the more that helicase broke the bonds, then it built number 2, then it built number 3. But this side is made in one continuous piece. It's just going to keep on going. The more it unzips, the more it builds. Okay, now I'm going to go back to the actual question. Okay. Explain why any newly synthesized strand of DNA is the result of both continuous and discontinuous DNA synthesis. How are those made? I would think here you could write about that they're both made by DNA polymerase. They're both made in the 5' to 3' direction, but the 5' to 3' direction goes in, on one side it'll go towards the fork, and on the other side it'll be going away from the fork. So the away from the fork side has to be discontinuous. I just want to see what they put for an answer for that. Circular, linear, perfect. Yeah, it's two coiled up. One long arrow on the lower strand pointing to the right. 3 arrows drawn on the upper strand and pointing to the left. From left to right, 1, 2, 3, perfect. Okay. The response indicates it because DNA synthesis only occurs in a 5' to 3' direction, synthesis of DNA strand. Oh yeah, that's all they wanted. That's all they were looking for, is that because DNA polymerase works 5 to 3, you're going to have one side that goes continually towards the fork and one side that goes in discontinuous pieces away from it. You can write all you want to there. You can write leading, lagging. You can include enzymes. Nothing is going to hurt you. That's what I like to say. Dump from your brain everything you remember about it. Don't spend too many minutes on it, but write all you can because it just shows the grader that you know what you're talking about. And for this question, you know what you are talking about, 100%. What else? Those were the 2 questions. What else do you want to go over? Oh my God, I wish there were 44 of you on here. It is recording though, I can't put it on there. Not really, I haven't even, not having a pretty hair day or self day. Ella, you're laughing at me? Anything else you want me to go over? So this is my plan. I am going to every week have free response questions that are going to be hopefully just like the ones that they're going to give you for the AP exam. I am going to post them both in AP Classroom and I'm going to put them in Schoology so that if you really like doing them like on a piece of paper or something, you can, or you can do it the other way. It'll be the same questions. I'm just going to pick questions from different units, whatever I find that I think looks like the kind of question they're going to ask you. That's really my goal, to have things that look like what the test is going to look like. And then each Thursday I'll go over them. I'll probably make up more than 2 though. Maybe I'll do like 4 a week or something. I don't know. It depends on what I find and how long I think it's going to take you. But it's a good time for you to like go over anything that you feel like you need extra help with. They are, if you go to the AP, what do they call that site? Not sure what they're calling it. But they are posting, I guess it's just the College Board site. The College Board site, if you type in AP Bio, there are review videos. I have seen some, they're a little bit boring, but maybe you'd be like, Merm, look in the mirror, yours is boring. I don't know. So I don't know. But maybe you just need at this point to hear someone else's voice. Like I've literally become like your mom in the background. So listen to somebody else because they're posting their videos and maybe theirs will say something that I haven't said. I'll put that link on Schoology because I know I can find it. I just can't think of what it's called, but I think it's just in College Board and it links you to different like YouTube lives that have been saved. Because in the beginning, they were only doing ones on evolution and ecology because those are what they said aren't going to be on the exam, but just to like sort of finish the year for you. But now I think they've gotten to the point that they're doing some review ones, or maybe May 1st, they're starting some review ones. What else do you want from me? What would be helpful? I might make a video of myself doing some free response questions that I find like, okay, when I read this, this is what I think of. This is what I think they're going to ask you for A. This is how I think you should write an answer. Sort of like what we just did. Does anyone now just want to go to like online college because you just want to do everything like this? No. Me neither. Me neither. I was looking through pictures and I saw this picture in my classroom and I literally was like, It was all you guys. Oh, and remember the day at the beginning when we were throwing the little balls and we went outside, like, how many can I make with my left hand and my right hand? Not the same. All right, are you muted because I muted you, or are you just all unmuted? You just did it. I didn't do it, right?
No.
You muted yourself. Okay. Anything else you want to say? Well, if you think of something and you're like, okay, I would be so much better off if I did this for you, please tell me, because I will do it. Whatever is going to help you feel like you're ready and prepared. Oh, everyone check to make sure they got the emails, right? You're getting emails from College Board? Okay. And did you show your parents that little, it's not really a PowerPoint, whatever it is that I made. I guess it's a PowerPoint.
Yeah.
Yeah. Yeah, I guess it's a PowerPoint. Someone else made it. I just changed it around. All right. Well, I guess that's it then.
Thank you. Okay,
Bye,
thank you. Thank
Yeah.
you. Bye. Bye.